Hi everyone. In this video, we will be discussing more about inheritance patterns and how different traits are um, inherited. So before we start, just take a moment to think, have you actually wondered how uh, offspring end up uh, resembling their parents, why siblings from the same parents are so different, how the transmission of traits end up occurring. If you thought of any of these, you are not alone. There used to be a scientist named Gregor Mendel. He was an Austrian monk who asked these very same questions. And at the time he was doing his work, he had two hypotheses where it first involved the blending part of the blending hypothesis, where they had equal contributions from both parents and that they basically mixed together. Then there was a particulate hypothesis where basically the two different uh, uh, discrete heritable traits basically have different units that end up being inherited as those specific units, and then they just like show up as it is. And what Mendel did was he wanted to figure out what was actually happening. He wanted to know which one of these theories of that time were correct, and he chose to use P plans to figure out how this all works. So when we look at Mendel's work, what we see is that he uses the common garden pea, or Pisum sativum. It is the first model organism in genetics. It's very easy to grow. It has a short reproductive cycle. It produces a large number of seeds, and the matings are easy to control. The traits are very easy in recognition. So Mendel specifically focused on 7 easily recognizable traits, which had 2 versions of each trait, including seed shape, seed color, pod shape, pod color, the flower and pod positions like these, or the stem length, depending on how long or short it was. And the different colors, just keep in mind as we go through this lecture. So what did Mendel actually do? Mendel followed the scientific method, basically keeping a hypothesis, observations, hypotheses, and just kind of went with his experiment, executed the experiment, and then see what his results were, and basically kept records of what he did, how he did it, very careful notes, and used that statistics to basically analyze his results. He presented his findings in 1865, but didn't even receive any recognition for his work at that time. But then later on, when scientists figured out more about like DNA and like how all of this works, his work ended up being rediscovered and became known as the founder of modern genetics. So before we get into some of the fun stuff in the ideas behind genetics and modern day genetics, we have to talk about some terminology, where the first set are genotypes and phenotypes, where basically there are heritable versions of a trait called alleles, and the diploid organisms that we are, that other organisms are, they basically inherit two allele copies, which make up the genotype for that trait. And in this case, it can be written like big F, or big F, little f, little f, and those will mean something in like a few seconds. So when the observed trait, so instead of like the genetic side of it, like what the genes themselves contain, the observed version of that trait is called a phenotype. So like if you have these two, it's like freckles for like big F, and big F, or no freckles with little f, and that just means that the uppercase F is dominant and the lowercase f is recessive. And if you have two copies of the same allele, like these two here, those are called homozygous. And then if you have two different alleles together, those are heterozygous. So this is like an example. It's like if it's like freckles versus no freckles, those might be dominant over the no freckles. And then this is another example where if you have like one dominant allele, it shows that characteristic more than the recessive phenotype. So it's just a matter of keeping track of what is a phenotype and what is a genotype. So now there are things called single gene traits, where if a trait is basically controlled by a single gene, it is said to be a single gene or a Mendelian trait. And basically other genes may influence the extent of these traits expressions, but you either have it in some form or you don't. It's one or the other. Where in, like I was saying earlier, in a classic dominant recessive genotype, having at least one copy of that dominant allele means that the trait will be expressed like in these two, like homozygous or heterozygous dominant traits. Now, the only way to express the recessive trait is if the person inherits two copies of that same recessive allele. So like these lowercase letters here. So now to kind of predict the whole probability of offsprings that inherit a specific genotype or a phenotype, there is a matrix that we all love to use, and it's called a Punnett square. So in one gene, the parents can only pass on one of the two gametes that they have. So either big W or little w in the case of like widow's peaks. So in this case, what happens is if you have both heterozygous ones, what happens is that all there is like 1/4 of a chance with the probabilities, except the heterozygous occurs twice. But when you look at phenotype wise, it's 75% widow's peak versus no widow's peak. So these are some activities you can try on your own. You can try putting your hands up, your thumbs like hands like this, or your arms crossed over your chest. Think about this and do this a couple of times and see which one actually lands uh, over yourselves. And what this kind of shows is does it happen the same way? If it does, these are some examples of like discrete heritable traits where there are two versions of a trait, left or right, but no in between. These are basically encoded by a single gene, but the degree of expression can be determined by additional genes or environments themselves. Now, to kind of figure out how the inheritance of these traits kind of occurred, Mendel used what is called a monohybrid cross, and he started off looking at the inheritance of one trait, which in this case was the seed color. They looked at it, and what they saw was with the Y allele, the big Y we'll see in this talk will be for the yellow seed, and the little y would be the green seed for being a recessive. And then, so these two genotypes are yellow, this one is green, and it's more dominant with the yellow, recessive with the green. Now, the first cross he did was between two pure true breeding plants. So one was purely yellow, so homozygous dominant, and then one was purely green, which was homozygous recessive. Now, when he crossed these plants, what happened was all of the offspring ended up with the yellow seeds. And these are your first generation pea plants. And Mendel was like, wait, did these traits blend in the first generation? Or is like the recessive gene or trait gone like forever? So what happened was he took that idea and he took it into a second cross between the first generation pea plants. So both of them were like the yellow ones, and what he saw was that he saw a 3-to-1 ratio in phenotype, or uh, a 3-to-1 ratio in phenotype. Is like, okay, so it did not disappear, and it's basically returning. So what happened is that these results kind of showed that it did not blend or disappear in that first generation, because they ended up reappearing in that second generation. And what happens is that the traits themselves are discrete units of information that can be transmitted intact from one generation to the next. So one fun question to ask is if you guys would ever dress like this for Halloween. It's like you can either dress up with your friends, saying a couple of them being peas and you being Mendel, or you can be like me, where I did not get my friends to dress up with me, so I just ended up having a sign with me saying "Lost Peas." Have y'all seen my peas? Okay. Anyways, um the what basically uh through this monohybrid cross, Mendel was able to basically develop the principle of segregation. This means that the alleles must be able to segregate or separate into different gamete cells during the formation of eggs and sperms in the parents, and only one allele of a pair can be passed on, and the offspring inherits one from each parent. So now that we know how one trait works, organisms themselves have many, many different traits. So Mendel's next question was like, do these traits end up getting passed on together like a package deal? Or is the inheritance of one set of alleles completely independent from each other, depending on the alleles you inherit for another trait? So, for example, if these two over here, like, let's call them Ellen and Bob, So Ellen and Bob both have freckles and widow's peaks. If they both have kids together, what traits will they inherit? Would they actually inherit all of them? So if they have 10 kids, what happens? So to kind of get the idea of this, um Mendel did a similar thing with his peas where his the two colors were looked into for the two traits, whether they were round or wrinkly. So now they when they crossed it, what happened was they saw that it came out to a 9 to 3 to 1 ratio when you have this traits together and cross together. So initially we talked about the principle of segregation, and seeing these ratios, what Mendel also developed is the principle of um, uh, independent assortment, where uh, each of the different traits can be independently assorted into different gametes, uh, independent of each other, and that all of the different combinations were allowed. So back to Ellen and Bob, where they showed that same pattern where they can have at least uh, the 9 kids would have the dominant traits, and then it kind of goes into the other phenotypes like these. But the one thing about these traits is that they were on separate genes and not separate genes, separate chromosomes, that they ended up um, being able to inherit independently. There are different ways that these wouldn't traits wouldn't be inherited independently, and we will talk about that in the next video. But to kind of have an idea of how to figure out the genotype of an organism that shows the dominant trait is to basically cross the unknown parent genotype with a recessive parent, with a known recessive parent. So if you have one that is unknown and one with recessive, and the progeny are all heterozygous or showing the dominant phenotype, what happens is that the parent itself is homozygous dominant. But then if you have a heterozygous parent with a recessive parent, you have half and half of the phenotypes. So that is what is a test cross, and keep in mind what outcomes that can occur, because in some cases you would actually use that instead of like other methods like PCR to kind of figure out what the genotype itself is. And with that, we will take it up with the extensions of these trends and patterns that we talked about today in the next video.